Hey, it's Jackson. I'm going to be going over the nitric oxide signaling pathway. This will hopefully be more of a review of the activity we did in class about the dog of erectile dysfunction, but still I'm going to go more in depth and hopefully explain it more clearly. So, G-Dog's dog had erectile dysfunction, and we can trace that back to a signaling pathway. Now, this is nitric oxide signaling pathway within an endothelial cell. As you can see on the left, the source of NO signaling is nitric oxide synthase acting on L-arginine. It's a complicated process that can start all the way from ACH signaling pathway. This pathway is both a paracrine and autocrine type of signaling, and we're going to be looking at how NO binds to its receptor, soluble guanine cyclase. But what is nitric oxide exactly? Nitric oxide is a gaseous signaling molecule that is made up of oxygen and nitrogen. It's mostly commonly known for its cellular response of vasodilation within endothelial cells, which is when blood vessels widen and allow for more blood flow. The nitric oxide signaling pathway begins when a cell receives a stimulus, such as from a neurotransmitter like ACH or even shear stress. ACH first binds to a G protein coupled receptor or GPCR. This activates the G protein subunit, and this GQ protein subunit acts as a secondary messenger to phospholipase C, causing it to activate. This causes PIF2 to convert into IP3 and DAG. IP3 diffuses across the smooth endoplasmic reticulum and is responsible for the release of calcium ions. These calcium ions then bind to the comodulin and form the calcium comodulin complex. This complex allows for nitrogen oxide synthase to be activated. But for nitrogen oxide synthase to synthesize NO, it requires arginine, oxygen, and NADPH. NOS converts L-arginine to citrulline by removing this nitrogen group, and this allows oxygen to convert into NO by using that nitrogen group from arginine. In this process, NADPH is also reduced into NADP+. It's important to note that there are two more types of NOH signaling. While I'm going to talk about eNOS or endothelial NOS, there's also neuronal and inducible NOS. NOS is found in neurons and facilitates neurotransmission, and INOS is found in immunocells and generates NO to kill pathogens. NO is able to directly diffuse across the smooth muscle cell membrane and activate soluble guanine cyclase, and this dephosphorates GTP to cGMP. The activated cGMP then causes the activation of protein kinase G or PKG. Finally, PKG phosphorates myosin light chain phosphatase, thus leading to the response of smooth muscle relaxation or vasodilation. One thing you might remember of the cell signaling pathways that we did with the broken dog is how Viagra was recommended to be used. Viagra works because in the cell signaling pathway, there's this enzyme called phosphodiesterase, which inhibits cGMP from sending a signal to PAG, which holds the nitric oxide signaling pathway. However, with Viagra, inhibits PDE from deactivating this pathway, which leads to the increased smooth muscle reaction. In other words, it causes penile erection. As you can see with vasodilation, it causes muscle cells to relax and blood vessels to expand, which allows for increased blood flow. This would hopefully allow your dog with erectile dysfunction to work again.